Thank you very much for inviting me. It's interesting that the previous two speakers gave themselves their own little setups and polling did the thing about the short attention span. And I thought, yes, I can use that because mine also will present just a few ideas. But whereas she presented things that were solved, I'm going to present things which we're working on, but we haven't solved. And then I realized Chris had a better way of putting it. So you say problems and opportunities. So that's the corporate side coming through there, that he knows how to do this. I'm learning, but I thought I would confess to you what's going on. So I'm from the European Bioinformatics Institute, and I have some of the corporate slides to tell you a bit about who we are and why we are involved in things that might be called big data. So it makes me laugh still, but there you go, we get things like this given to us. So, revolution. I read an article in a magazine once. It was a gardening magazine, and it says never buy any product that's described as revolutionary. And we couldn't decide whether that meant like revolutionary new or actually, you know, in the garden things that revolve are not good. So the revolution is coming about from our ability to understand, well, other things as well now, but you know, this change that we can do DNA, genome sequencing, so rapidly, so easily, so inexpensively. Plus, of course, the important factor, which is like essentially everything is DNA or is made of DNA. All the important things we need to know have, at least at their heart, many factors depending on the genomes, and that's something we have now got access to understand what's going on, whether we're talking about populations or bacteria or what's going on in the sea, or, you know, all of agriculture is about trying to get things to grow, to feed us, that are all controlled by their genomes, or sampling the environment, or any of that stuff. And what's changed, people have said, but I have the picture, you know, what's changed is a sequencing device has gone from being an institute to a laboratory to a big box on a desk with a computer underneath to a little thing that you hold in your hand that plugs into your laptop computer, and coming later this year, they claim, or in the near future, the thing plugs into your phone, not your laptop computer. So it's making it faster, smaller, cheaper. It's bringing it to thousands more people. And instead of being an institute making a decision to sequence a genome, you decide which part of your lunch to sequence to check it's what it's meant to be, or is it infected with bad viruses, or, you know, is it really made of horse, or whatever it is you're worried about this week. And so that was the genome sequencing revolution is happening, is still going on. And more stuff's coming at European Molecular Biology Laboratory, my parent organisation. We're world leaders in imaging of cells. So just you light microscopy to a greater and greater resolution to be able to see more and more of the stuff that goes on in living cells, every cell's down to the level of individual cells, and looking inside individual cells and seeing biological processes going on. So now we are just bracing ourselves for a flood of image data to be coming in as well. We heard a little bit about that earlier too. And why are we worried about it? Well, it's because European Bioinformatics Institute is Europe's home for the public domain molecular biology data. So this is the genome sequences, the protein structure information, and we think in the near future we're going to be asked to do the same for a vast amount of cellular imaging data. And there's a lot being held by companies and that's not public. There's a lot in health records that you can't readily go around storing and making available to the scientific community because of security issues and confidentiality issues. But all the other stuff, all the open stuff, and we modestly say we're Europe's home, but we're one of the two global centres for storing this kind of information, the other one being the US. We're based just south of Cambridge, next door to the Sanger Institute, for those that know the Genome Campus. And we're part of the MBL, the European Molecular Biology Laboratory, which is a sort of international government-level subscription, join-in collaborative science project, kind of like CERN is for particle physics, and we're that for molecular biology. We're not funded as well as them, sadly. And we are seen as one of the global trusted places to deposit this data, to try and do something more useful with it than simply archive it, but to try and integrate all the different sources of data and add value and knowledge around that, but then to serve that back to the research community, to companies, but to individuals, to just anyone. It's entirely public. So we have a large data center. We currently, the headline figure is 120 petabytes of storage in use, more than 40,000 CPU cores kind of serving all this, fast internet connections and the like. And this, without going into any detail, is an idea of the kind of the range of data sources that come to us that we're meant to store, archive, serve back, cross-reference, serve back the results of that, and all as resources for the research community in many different molecular biology areas. And this is the kind of idea they provide me. People send us data, we archive it, we classify it, we share it with others, we integrate, add value, provide it back to researchers, and hopefully that's a progressive, a loop of information being shared that's advantageous to all. And I recently got told to add this one in, which is that we subscribe to the fair data principles, which is not just the word fair, but there's a kind of movement now about making sure that open data is made maximally valuable to other researchers, and there's some general principles about making sure people can find it, they can get the data out, you move it from one process to another, and they can reuse it and make the results reusable for others too. That's what we do. This is the obligatory slide about how we get more and more data over time. So the x-axis is time on a linear scale, and the y-axis is the number of bytes of information we're storing in some of each line represents a collection of databases in different areas. And being on a logarithmic scale, it just means basically it's going up exponentially on the whole. It's certainly, even with improvements in the amount of computer storage and processors you can buy per dollar or pound of your euro of your funding, we're falling behind the curve. Our funding does not increase. Our funding plus improvements in computers does not keep up with the growth of data, and periodically we go through angst about whether we can keep on storing all the stuff people expect us to store. So what I asked some of my colleagues to tell me about data scalability issues, and I've got 3 small stories I'm going to try and get through in the rest of the talk, just to show what some of the problems are, and that we've got some idea about how to deal with them, but we're certainly always on the lookout for help, collaboration, friendly people who will let us come and bend their ear about specific problems we've got, and so on. So maybe that's the value of me being here today. So I want to say something about the UniProt database is this repository of, we like to think, knowledge about the structure and sequence and function interactions that proteins and protein families undergo. Proteins are the things that get made. The message in your genome tells the cells what proteins to make. The proteins undergo chemical reactions in your cells, and lo and behold, that's a living organism. So it's vitally important. So the left-hand side kind of symbolically represents all this data that scientists out there are generating. They do their own experiments, they get raw data, they get the results of other things, and they have their own uses for that. They're all doing it for a reason, but one of the byproducts is they send those many different things to us. And they say, you're the global archive of this, just store this for me and make it available to everyone else in the whole world, please. And while you're at it, there's 27,000 other things each one relates to. Could you just find all those and cross-reference them and make that available too? And we say, yep, okay, we can do that. We'll have a system with cogs in the middle of the diagram. The red ones are kind of just cleaning up the data they send us, because they don't all send it in the right format or a consistent format. So there's various processes we go through to kind of make it analyzable by a centralized system. And the purple ones are actually doing something with it. We're trying to understand what's in there, work out the cross-referencing, work out which things that arrived today are related to which things we had from 4 years ago, and how useful is that. And then that's all put into what we call a knowledge base, but this is this collective archive value added integrated database. I mean, it's way simplistic to call it a database, but conceptually It's this huge storage of all this information. And so UniProt does this for the world of understanding proteins. And so their own version of the data is growing and growing. Comes on a slide that looks something like this, that shows how they started in 2002 when these records on this one started. There's something like 1 million proteins out there that we are being asked to store information and integrate and share that. And then in 4-year periods, it goes up to 3 to 10 to 30 to currently something over 100 million proteins that people know something about from some experiment and they want something to do with. I've forgotten the figures now. What have I got? It's doubling every 26. The green, which is like the raw things that have come in that we're trying to deal with, doubles every 26 months or so. And then the 3 color schemes, green is like what we would like to be able to deal with, blue is what we maybe can deal with, and the red is the really, that's the ones where humans have really looked at this and done this by hand, and we have a very, very good understanding. We can't do that for the whole lot. That's completely infeasible. So a lot of the work now is what computational methods do we have to make the connections across 108, 107 million records in the database. And the other thing I've got to remember to say at this point is that's not 107 million data points that we're aiming for. That's 107 million databases, because each protein has a database of information in it. We know its sequence, we know what organism it's from, we know what other things it's been proven to react with or not react with. So you've got to try and create something over 100 million databases and then make those usable to people outside. And it's difficult, but over the years it's grown, and we're kind of capable of doing at least some of it represented like this. And what we have here is a big funnel, and everything gets churned through. But the point of showing it is, the little labeling at the top is some of the things we are trying to deal with, all of which will be affecting how fast we can do this and how accurately we can do this, and its value. And we could have picked many of these to try and show that there has been some sort of progress. And the one I chose, because of who I asked, I think, who's in the audience, is something about the association rule. So this is the idea that we now have knowledge about a subset where humans have checked and looked into it, read the literature, understand what the proteins are doing, and we're trying to find rules that enable us to put the same kind of annotation on other sequences that haven't had a human looking at them, but which arrive regularly uploaded to the databases. And I'm not going to explain how it's done because I don't know, though there's an expert with a poster this afternoon who can tell you more about it, but Poling's was better. But a load of maths happens, which is indicated on the right-hand side, that has theorems, results about how we can most reliably transmit information which we know about one protein and we know something else about another one. Can we say this fact applies to that one with some confidence? And one of the things there has been work on in recent years within this database team and with others is to find more efficient ways of doing that so that we can more quickly and more robustly share the information we have across other proteins. And we have some progress to do that. That's one. Got 2 more, 15 minutes, okay. Should be fine. Right. That's the chance for each one of us in this room to get cancer at some stage in our lifetime. And that's the percentage of us in this room who will die of cancer. Cancer is a very, very big deal. Obviously, we've had it mentioned already today. There's a huge amount of research going on to try and understand this. Some of the causes, there's different ways. There's very large projects going on globally around the world working together to try and understand it. There's major rearrangements of the genetic material, the genomic, the chromosomal material in your genome, which is one direction that's greatly accelerated in cancer cases and is obviously a massive cause and problem. And then there's much smaller, like what if you just have one little mutation at one position in a protein that stops the protein working? You don't need to rearrange your whole genome to break the function of certain proteins. It can be a tiny change in one letter of this alphabet, and I don't know if you spotted it, but there's one red one in there. But that's an easy example. They're not nearly that easy to find. Because for a computer, it's not that hard. And we've got to study a very, very large number of individuals and cancers and cancer types and different tumours from people. The idea of this is to show that how frequently do you get the same mutation in different cases. So it's just, so the most common small mutation causing cancer is the one on the left-hand side that's up around the 30%. You see that. It's not the only one, but you see that in over 30% of people with cancer. But that very rapidly drops, and you've got a stupidly long tail on this distribution. Most mutations only occur in a very small number of people. So you need to study a lot of people to see the set of possible mutations and to understand all the different things that are going on. So it's become a global project to sequence thousands of people with cancer, both their healthy genome and also the genome in cancer-affected cells, before we even get on to doing every single individual cell in those cancer tumours and so on, which is coming too. I mean, not every cell of every tumour, but if you could do that, you'd already solve the problem. But if you could get them out, but you know, to this in-depth understanding. So the pan-cancer analysis of whole genomes is a part of the global cancer collaborative project. Doing that, they've spread the work across many groups, and they've identified 39 cancer types to look at. They've ended up with almost 3,000 tumor types in their data set, 300 scientists collaborating, looking at 650 terabytes of raw data. And one part of the process is to go through looking for these mutations and recording them, classifying that, and then that goes into further downstream analysis. But it made a nice example, I felt, because they had this graph. And actually, this problem that they have just scales linearly, in fact, because for each sample you look at the moment, it's a patient, and you've got their genome, and you've got a genome of some cancerous cell from that person. And if you get 10 times as many people, you just have to do that 10 times as many times, and that goes downstream. So actually, it scales not that badly, they thought, and so they set up their pipelines to analyze these 3,000 individuals' data that were going to come through, 300 tumors. And the way it was told to me was, and they have different parts of the pipeline. So the first part of the analysis is this thing where you take the raw data and you compare that from the cancer cell and the healthy genome for an individual, and you work out where all the differences are. And because of the way the sequencing is done these days, it's not, you don't just get presented with 3 billion letters twice and you're looking for the differences. It's a lot harder. It's the slowest step in the process for what goes on. And that's the line on the left called BWA. And this is over the actual time on the x-axis of the project, which parts of the analysis was the consortium of researchers able to run. So that was the first analysis, and then it was, once the results become available, they go off to different institutes, the Sanger Institute, DKFZ and EMBL in Germany, and the Broad on the east coast of the USA, to go and do their thing to it. So they set the pipelines up and they set it all running, and over back there in 2014 they thought, this is fantastic, it's going up nice and straight, we're racing through this. They got about a year in, they thought, we've got about 3 months to go and it's all going to be done and then we can all move on, and the other group started. And then it all flattened off and it crawled through the remaining cases. So when they set this running, most things work, they get the raw data and they start analysing it, occasionally it fails, and then they resubmit it, something's gone wrong somewhere in the system. And they do that, and it was described to me, they said, well, maybe we were a bit naive about the way it was going wrong, because we just did that for a year, and then everything kind of ground to a halt. So by a year or whatever it is in, the analysis at the Sanger Institute had started, and they found the same. They were able to start off at great speed doing what came next for them, and then, they got, even when there were still samples to go, they had the same effect, that it kind of slowed down. And all the analyses had the same thing. I was also, before anyone asks, the big dips in the blood, one person told me that was just, he didn't make the graphics and he thought that was just a problem with plotting the numbers. But I was at a different talk yesterday and someone else was saying actually they got a whole load of their analysis wrong, so it kind of went up and then they had to withdraw the results. So it is actually possible for the curves to go down again. So I think that's some of what's going on there, but it's a different problem. So they looked at what was going on and they said, well, this shouldn't have been a problem. Everything started off just fine. Why did it go wrong? Why did it not scale linearly for us? Why did we spend all this time kind of having to manually fix things? So this is, they learned. So they, things went wrong, and it became an engineering problem. They said it wasn't a problem of let's be genius statisticians or mathematicians and change the problem to one we can handle better. It's why was the method that ought to have worked just fine not working, and what could they do about it? So there's obvious, hardware failures, there's bioinformatics failures, that the analysis is not good, or the data is not good and needed cleaning up. And they ended up realizing that what was their bottleneck was not the size of the problem, but the manual investigation they were doing, because they got a year and a half into the project thinking everything was great, and then it all ground to a halt when they were faced with a series of problems of things that wouldn't work. And so what they were able to do was devise a much cleverer pipeline. They realized that it's the cases that were failing that was the problem, and they were being very naive about this. If something failed, they kind of just resubmitted it to the queue, and they ended up with they'd done all the ones that worked, and all they had was a backlog of cases that didn't work because something was wrong with the data, or it was an edge case and the analysis couldn't cope with it and they had to tweak it. And then the bottleneck was humans trying to resolve each of these problems. And they then devised a new pipeline that was much cleverer about saying if there was a problem, to try and work out what it was, and to work out if a human was needed to intervene and change something and resubmit it. And they worked out that the left-hand graph is how much of the study was complete over time in the different parts of the project as it was. So presented a different way, it's the same data as on the previous graph. And with the new clever system that they now have available, which is called Butler, they can kind of do it all in linear time. They identify the difficult cases consistently over time, and they can smooth the whole thing out. So by solving an engineering problem about working out when things didn't work readily, they were able to recover the kind of performance they ought to have been able to get in something that scaled in a straightforward manner that they hadn't imagined would be difficult. And the last example is more of a classic one. It's the idea of trying to associate changes or differences in the genome, variants between one individual and another, and the phenotype, which means some measurable characteristic, like do you have diabetes, or are you overweight, or what is your height? And how can we find which parts of the genome are responsible for some of these different features? And the basic strategy is you measure a bunch of individuals and you look at their genome, which is the rows on the top right little graphic, and you look over different people, which is, the genome is the columns, and the individuals are the rows, and you look where there are differences, and you try and relate where those are differences to different things you know about the samples, like which ones have diabetes or don't have diabetes, what the height of an individual is, are they obese or not, and so on. And it's a really large-scale testing problem, because you start off with, maybe you're looking at a genome region of 10 to the 6 long, or in fact probably in the whole genome we're looking at 10 to the 6 variants, and it might be any one of those is responsible. So that would be relatively simple to find which one of those where there's changes does it explain diabetes status. But it might not be one, it won't be as simple as one. There'll be many factors that explain diabetes, and they'll be in combination, and there's an astronomically large number of possible things that could be the explanation. How do you find the right one? And there are models and methods for trying to do this, and they don't work terribly well, and you need a huge amount of data to debug them and work out what's going on in difficult cases. So we've now got problems where we've got something like, a data matrix of 10 to the 6 columns by 10 to the 5 rows to explain something from 10 up to maybe 10 to the power 5 phenotypes, the different measurements you might be taking. So 10 to the 5 isn't going to the doctor and they record 10 to the power 5 factors about you, but a lot of automated experiments now will come up with when a certain chemical is introduced to your cells, what happens, and that can be done as a massive screening exercise, potentially over multiple patients. So how do we explain maybe 10 to the 5 measurements where we have variation in 10 to the 5 people, which can have 10 to the 6 components to what's varying? It's a very, very, very big problem. I mean, trivial things like large scale, it's a multiple testing problem. I mean, that's, you've got to get a long way to even reach the fact of worrying about how to deal with that. This is not straightforward data to analyze. And there are ways of doing it. One of Oliver Stegler, who works at the EBI, is expert in devising these sorts of methods and trying to understand what's going on. And what he's realized with one of his PhD students recently is that there's a lot of structure in this, and that is where you're allowed to speed things up. So he's got to do this massive data matrix modeling of what's going on with matrices of the size, those numbers I was saying before, dimensions 10 to the 5 and 10 to the 6, in a model something like the one at the bottom. But there's a lot of structure in some parts of it. So for example, amongst your 10 to the 5 individuals, they're not different, random samples from some homogeneous population. There will be degrees of relationship with them. If you're looking in one community, they might be relatively closely related. If you're looking across the nation, you will have a series of closely related or relatively closely related. And this gives some structure that you can use with luck to try and speed up some of the work that goes on. So essentially, big matrix factorization problem, is there some structure in the matrix which we can use to help us do it faster and retain accuracy in doing that? And the answer they discovered for this one, for example, was, so in the kind of modeling they were doing before, the sort of naive analysis is it takes n cubed time as the number of individuals, so different patients who've been studied, goes up, you get a power 3 relationship with how long it takes to do the matrix analysis. That's kind of pretty hopeless. So this graph, it's CPU time for some standardized study analysis on a log scale, and also on a log scale, the x-axis is the number of individuals you might be dealing with. And this whole range up to the far right is relevant. At the right-hand end, we're talking about 500,000 individuals. That's the kind of place we're going to be, if not now, then in a few years in UK Biobank, 100,000 Genomes Project, and other countries doing the same. So it's not one of those studies where we're living over in the left-hand edge and everything is okay. We've got all of this to deal with. N cubed wasn't good enough. They were looking for ways of improving that. They were able to use this structure in the matrix that described the relationship amongst the individuals to get an acceleration, which is the blue line, which comes in somewhere n to the power 2 and a bit, or it's even a bit better than that. Maybe it's just under 2 towards the end. That was much better and still not good enough for what they wanted to be able to present. And then with an additional, this was what if the matrix isn't as neatly partitioned as we hoped it was for the blue analysis, but can we make an approximation by assuming it is? So the result is no longer as accurate as you wanted, but you can get it faster, and then they can get it down to approximately n. So now it's gone linear, and now there's sort of hope that at least, I think when things are linear it's good, because if you can do it per patient, then you can kind of justify the resources to spend that. If you say, well, now if we're going to have 100,000 patients being tested for a certain thing or studied for that now, you kind of understand if it's going to cost 100,000 times as much, including if you're asking for 100,000 times as much computing. If you say, well, we're going to do 100,000 people, but it's going to take 10 million times as much resources, then you say, then each person is now being asked for whatever the ratio of those was, 1,000 times as much money from their tax bill or whatever it is. So this was seen as a success. These are the kind of problems we have. They get even bigger. There will be more and more measurements made. This is the 10 to the 5 phenotype kind of measurements, which is you might be looking at people's genome, you might be looking at the protein products going on of reactions happening in individual cells, which will be different in different individuals, but different parts of the body, different tissue types, different times of day, the weather will all affect these. These are all now going to become standardized assays that your doctor might do anytime you go and see your GP. In the future, metabolomics, what are the reactions going on? How are you digesting your food? What are your sugar levels like? Will all become, so the number, the problem only gets bigger as we go on. That was all I wanted to say, so I'm more or less on time. I'll get out to lunch. My thanks to my colleagues who provided me with the examples. Thank you very much for listening.
I think I'd like to thank both Nick and Chris for two fascinating talks. We are back on time, and I think we should celebrate that by going for lunch. Obviously, luncheon posters between now and 2 o'clock, and if you have any questions for Chris or Nick, then please take them up over lunch.